Moving forward, researchers struggled with what the driving force of the brain was. We know it wasn't animal spirits, it wasn't water, it wasn't air, it wasn't special vibrations. The first clue came from an Italian physician, Luigi Galvani. He discovered animal electricity, and he did so through touching frog legs with electric sparks, so experimentation on frogs. And when he did so, the frog leg would contract, and that showed that electricity passed through the nerves of the frog leg. Again, with frogs, this was common at the time to do experimentation on frogs. This German physician also provided evidence that electricity caused the transmission of nerve signals and discovered what we now call action potentials. Hermann von Helmholtz, a German physicist and physician, was an incredibly talented scientist. For our purposes, in this class, he's credited with finding out the speed of a nerve impulse. Most people thought that it was pretty much instantaneous once electricity kind of came to mind, and a lot of researchers, but he found it to be much slower, around 25 meters per second. But overall, these three scientists really solidified this idea that electricity was the driving force in brain function, not animal spirits, not air, water, or vibrations. Moving right along past these forces, the big thing to investigate and discover now was brain function and localization of function. This is best described through the pseudoscience of phrenology. Franz Gall, a German anatomist and physiologist, became famous for developing this field of phrenology, which was the process of correlating bumps on the skull with different mental faculties. He thought that pronounced areas of the brain would be reflected through the non-uniformities in someone's skull. So through this, theoretically, profiles were developed of people about their abilities to deal with other people, their memories, how stable their emotions were, all from just the shapes of their skull. This pseudoscience was pretty wildly popular for a while, but it did eventually fall off, and that's because just the science was fundamentally flawed. You definitely cannot tell anything about a person from just the shape of their skull. But yet this idea of localization of function was kind of right, that there were specific parts of the brain, not the skull, the brain, that were responsible for various behaviors. So at this time, the biggest debate now was whether localization of function was really a thing or not. A big advocate against localization of function was JeanPierre Florey, a French physiologist. He very meticulously experimented on animals. He would ablate various parts of the brain. And ablation here means just the destruction or removal of a part of the brain, whether it be from natural causes, brain damage, or a surgical scalpel. Anyway, he would ablate various parts of the brain and then track different behaviors of animals post-surgery. From his experiments, he noted that aspects like human intelligence or animal intelligence or intelligence in general, it was more of a unitary function, not belonging to any one region of the brain. So through actual experimental evidence, he was a pretty strong critic against this localization of function idea and obviously the pseudoscience of phrenology. But then came Paul Broca. Broca, with these beautiful sideburns here, was a French physician who studied a very particular patient who was known as Tan. This was because this patient, Tan, lost the ability to speak and could only utter repetitive instances of Tan, Tan, Tan. A lot of people, I think, say Tan, but I think it was actually pronounced Tan. Anyway, after this patient passed away, an autopsy found only one region of the brain where the brain tissue had died, or a case of natural ablation. This was the left frontal cortex, and we call that area Broca's area now. So Paul Broca therefore deduced that this part of the brain was responsible for this particular deficit, and therefore the production of speech. And this actually gave some type of validation for a phrenology type of science. So not the skull part, but more of the localization of function part. More evidence now from Karl Wernicke, a German physician discovered and called Wernicke's area, which was in the left temporal parietal area, and that's highlighted in blue here. This area is responsible for speech comprehension after his patients who had the ability to produce speech. but would just utter strings of non-comprehensible sentences. So everything would be just jarbled or garbled up. If you YouTube Wernicke's area or Wernicke's aphasia, you'll be able to find some pretty interesting examples of it. But the overall idea here is that some type of localization of function was winning out in the field of science. In today's knowledge, we know that there are particular regions that are really involved in specific functions, but no region only does one thing. There are whole networks of distributed brain regions linked to various functions, but that's just a side note. We'll talk about brain networks later on in the course. The last piece of the puzzle I want to touch on is neurons. Cell theory was huge for neuroscience because it detailed that living organisms are made of cells and that these cells are the fundamental units of biological organisms. This Czech physiologist, Perkanji, actually discovered some of these fundamental units, some relatively large cells in the cerebellum. And we call these large cells in the cerebellum Perkanji cells today. And it was amazing because now neuroscientists were beginning to be able to visualize the actual structure of neurons and just to see the characteristics of them, see how arboreal they are, kind of tree-like with extending branches. Our ability to further visualize individual neurons came from Golgi, who was known for the Golgi stain technique, which is a way to stain and isolate individual neurons for visibility. Building off that technique, a Spanish neuroscientist, Ramon Cajal, used this Golgi staining technique and actually built off that and made a pretty unique discovery that neurons were not continuously connected. So before that, everyone just thought that neurons were physically connected to one another, that they were continuous. But Cajal was able to see that there were actually breaks between the neurons, and we call them synapses today. But the idea that nerve cells were discontinuous was revolutionary at the time. So the work of both Golgi and Cajal led them to both win and share a Nobel Peace Prize. Not a Peace Prize, sorry, that's a mix-up. Just a Nobel Prize of Science. Okay, so anyway, I will wrap up, but I just wanted to give you a glimpse of how we got here. The fact that we started out debating whether the brain was the important organ after all, then discovering the driving force of the brain, that it's not these special animal spirits, but electricity and chemistry. Then we narrowed in on what the brain does and what it is responsible for, this kind of localization of function to some degree. Then we learned what the fundamental unit of the brain was with cell theory. From there, we were just off to the races in brain sciences. So if you do want to learn the complete story, replete with many more pioneering scientists of the time that weren't described here, The Idea of the Brain by Matthew Cobb is just a brilliant book. So if you have some time, I would definitely check that one out.